Many things can go wrong when a gene gets switched on at the wrong time or place. New research by Doctors Gruel and Falco in CCR's Laboratory of Biochemistry and Molecular Biology shows that when a gene whose activity should be reserved for producing sex cells becomes active, cells can't properly sort their chromosomes when they divide. This
untimely expression of gametogenic genes in vegetative cells causes uniparental disomy.
Most of the cells in our bodies, called somatic cells, contain two sets of chromosomes, one maternal set inherited from mom and one paternal set inherited from dad. Errors in chromosome inheritance can lead to abnormalities. For example, UPD occurs when a cell receives both copies of a particular chromosome from the same parent, instead of inheriting one copy of the paired chromosome from mom and one from dad as they should. Initially proposed by Eric Engel in 1980, UPD is a frequent chromosomal anomaly with an incidence of 1 in 3,500 people and has been linked to cancer and numerous congenital diseases. Despite its clinical importance, the molecular factors that trigger UPD have remained unknown. Because cells that are affected by UPD contain the same number of chromosomes as normal cells, the phenomenon is not easy to study and detect in humans. However, by conducting experiments in fission yeast, Doctors Gruel and Falco were able to detect UPD, which led to the discovery of the first causative factors ever identified for UPD. The fission yeast genome only has 3 chromosomes, yet it shares many features of chromosome structure and gene regulation with human cells, making it a powerful genetic model. One advantage of this model is that they could employ genetic markers that produce a color readout for UPD detection. As shown in this picture, UPD has occurred in the red and pink yeast colonies, but not in the white ones. Many years ago, the research team had previously discovered that a gene-regulating system called RNA interference, or RNAi, controls the DNA packaging inside cells. They also noted that defects in the RNAi system can cause cells to make mistakes in segregating chromosomes during cell division. By using genetic assays to detect UPD, the research team established that UPD occurs at high frequency in RNAi-deficient cells. Interestingly, they had previously discovered that RNAi prevents the expression of genes involved in gametogenesis, the specialized process that gives rise to eggs and sperm. Gametogenesis should only occur in reproductive cells, so these genes are kept tightly silenced in the somatic cells by RNAi machinery. After analyzing 27 abnormally expressed gametogenic genes, the research team found that only 2 genes were linked to UPD. Doctors Gruel and Falco are looking to begin studying small molecules which can interrupt UPD in fission yeast, such as drugs that disrupt gametogenic gene expression, which can eventually be tested in human cells as a potential therapy or treatment option.
Storyboard and script by Doctors Shiv Gruel, Diego Falco, and Jemima Barrowman. Illustration, voice, and editing by Jennifer Wang.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4CANCER. Produced September 2018.